So students, in today's class, we are going to learn about polymerase chain reaction. So the aim of the experiment is to amplify a specific DNA fragment by using PCR or polymerase chain reaction. The requirements for the experiment include template DNA, the enzyme Taq DNA polymerase, including all its buffer components required for the activity of the enzyme, forward and reverse primers, VNTP mix, and nuclease-free water. And in instruments, we also require the equipment that is thermal cycler and gel electrophoresis setup and trans illuminator to observe our results. So coming to the principle, we have the first step is known as initial denaturation. The commonly used temperature for initial denaturation is around 94 to 98 degrees Celsius, depending on which polymerase enzyme we are using in the for the extension reaction. And the time frame for which we perform the denaturation is generally 1 minute to 10 minutes time frame. Then following so initial denaturation is basically for separating your double-stranded DNA into single-stranded DNA so that it is accessible for the primers to bind. Then we have the 3 steps, that is denaturation, annealing and extension, which are repeated in a cyclic fashion several times. Approximately 20 to 30 cycles are commonly used. So during the denaturation step, again your DNA denatures and the temperature used for denaturation is same as that we use for initial denaturation, either 94 or 98 degrees Celsius. Time frame is around 30 seconds when we use the denaturation time. And then we use the next step, that is annealing, which is generally set at 55 degrees Celsius. However, you can sometimes modify it to bring it to as low as less than 50 degrees, around 48 degrees, or as high as 55 degrees Celsius, 59 degrees Celsius also occasionally if the PCR product is not amplified. So at this temperature, your primer binding will take place to the template at specific DNA sequences. Then the next step is extension, and the temperature that we make use of here is around 72 degrees Celsius, and the extension time again depends on the size of the DNA fragment and the type of enzyme that we make use of. So during this step, the enzyme will bind at the 3' free 3' hydroxyl end of the primers and will carry out extension in 5' to 3' direction, leading to synthesis of the daughter DNA strand. So here in PCR, we can make use of up to 30 cycles commonly, and with each cycle there is doubling in the number of products from 2 to 4, 4 to 8, and so on. So this is basically an exponential amplification reaction. Then we have a final extension step, which is again at the same temperature that we use for extension, around 72 degrees Celsius, for about 10 minutes, and this provides extra time period to complete any unamplified unit. Then we have the final hold temperature, which is around 4 to 10 degrees Celsius, and here we are basically lowering the temperature in order to stop the PCR reaction. And so after this, we can remove our tube anytime by switching off the instrument. So all the points that we discussed are written here for your reference in the 2 slides. Then coming to the procedure, the first step is preparation of a PCR master mix, which is set up by adding all these different ingredients. So the first component that we add is nuclease-free water, which is maximum in volume. And then we add the rest of the ingredients as per the mentioned volumes. So your assay buffer basically contains stress HCl buffer that is required for maintaining the pH around 8 to 9 required for the enzyme activity. And it also contains potassium ions that are required for proper primer annealing, and magnesium ions that are required for the enzyme activity. Then the template DNA is basically a source of information that contains the gene sequence that is to be amplified. And the primers, forward and the reverse primers, they basically define which gene sequences are to be amplified. Then we have the DNTP mix that contains all the four building blocks, that is ATP, CTP, GTP, and PTP, which are required for polymerization of the product DNA. Then we have the enzyme that will perform the actual amplification reaction. So two types of enzymes are commonly in use, either the tag DNA polymerase or the other type of enzyme that can be used is fusion DNA polymerase. So when we use tag DNA polymerase, the amplification or the extension time is about 1 minute per kilo base pair. And with the fusion DNA polymerase, we use 1 minute per 30 seconds for extension. And the initial denaturation temperature with tag DNA polymerase is about 94 degrees, while with fusion DNA polymerase, it is around 98 degrees. So once we set up the final reaction, we then proceed to the next step. So we have also the roles of all the different components that we just discussed are being mentioned in the two slides. And then coming to the next step is we place the reaction that is present in the PCR tube in the thermal cycler, and we set up the PCR program. So we have initial denaturation at 94 degrees Celsius for 10 minutes. And then we have 30 cycles of the following three steps: denaturation at 94 for 30 seconds, annealing at 55 for 30 seconds, and extension is at 72 again for 30 seconds, which means the fragment is approximately 500 base pairs in length. We have a final extension temperature at 72 degrees Celsius for 10 minutes, and the final hold temperature, which is at around 4 degrees Celsius. Following completion of the PCR reaction in the thermal cycler, we then analyze our product by performing agarose gel electrophoresis, and we interpret the results by observing the gel in a gel dock. So here are our results, and the ladder that we have used over here is a 100 base pair ladder. So this is a reference 100 base pair ladder, and the size of each fragments are about 100 base pairs apart from each other here. So you will be able to identify the difference in smaller DNA fragments. So as you can see, the PCR product that is amplified is around 400 base pairs in length. So that's it about PCR. Kindly like, share, and subscribe if you like the content. Thank you.